Good evening. I'm Matthew Moss. I head the Development Office at Homerton College. The Development Office is responsible, amongst other things, for keeping alumni connected with the college. So a very warm welcome to this year's virtual alumni reunion weekend. If this is your first event of the weekend, then you're doubly welcome. So every year as part of Alumni Reunion Weekend, we host an academic research talk, and we spend traditionally some time scratching our heads and wondering what the topic ought to be. We didn't scratch our heads for very long this year before deciding on our topic. We've all suddenly developed a boundless interest in virology. Homerton is in a great position to talk about virology and contribute to those conversations. We have two standout virologists among our fellowship. Professor Ravi Gupta joined Homerton last year, and this evening's speaker is Dr. Julia Kenyon. Julia joined Homerton as a fellow in 2010, when she also became Director of Studies in Biological Natural Sciences. She's been a tutor for our students since 2014, and she additionally took on the important role of Director of Studies for our first cohorts of preclinical medical students who joined us as our first medics in 2015. Julia's research as an independent scientist in the Department of Medicine at Cambridge focuses on how viral genomes are structured. She researches HIV, FIV, the feline equivalent, dengue virus, and now the novel coronavirus, SARS-CoV-2, which causes COVID-19. Julia also co-directs a laboratory in Singapore, and so her Skype and Zoom skills predate most of ours, and certainly mine. Julia will talk to us this evening under the topic COVID-19, what can we learn from other viruses? And she'll take questions at the end. The best way to ask a question, if you want to, as the presentation goes on, is simply to type it in the chat. If you look at the bottom of your screen on most ways of accessing Zoom, you'll see a chat button. If you type questions in the chat as we go along, we can pick those up at the end and make sure that they're covered. It gives me a very great pleasure indeed to introduce Dr. Julia Kenyon.
Brilliant, thanks so much. And I'm just trying to work out how to share my correct desktop. There we go. Is that coming up okay for everyone? And can you hear me okay?
Yes, that's fine.
Great, okay. So hi everyone, I've just had a quick look at the attendee list. I realised that I've got one or two of my ex-students, my ex-tutees in. Hello. It's really nice to sort of be in contact with you. You can obviously see me, I can't see you, but let's have a quick email or something afterwards anyway, and let me know how you're going. So today I'm going to tell you about COVID-19 and what we learn from other viruses, obviously. So we're going to go through what a virus is, how or why we get a new human virus, how or why viruses cause disease, how they spread, how we can stop them from spreading, and then I'm going to tell you a little bit about our research that we're doing. So a virus is a parasite that has to live inside cells. And it has a genome, which is DNA or RNA, wrapped in a coat that's made of protein. So sometimes it will have a lipid envelope on the outside of it. That makes it a bit easier to destroy by cleaning products and things. And we're lucky that SARS-CoV-2 has one of these. This is my diagram of a virus, and that's showing you why I am a scientist and not an artist. And this is how they then replicate. So our virus enters a cell, and it disassembles itself into its various components. They get replicated, it puts itself back together. All the time it's using lots of different cellular components in order to do these, and then it exits the cell. And it may or may not destroy the host cell when it does that. So this guy is Charles Darwin, appearing here as the original hipster. And this demonstrates his voyage around the Galapagos Islands, where he popularized the theory of evolution. And so here are the finches that are resident on these different islands. And when we have species that are geographically isolated, then those species have environmental pressures put on them, they have different food sources, and so over time they evolve to adapt to the conditions that they're in, and you can see that here in the finches with those particular beaks. So evolution depends on the genetic material. Most cases that's DNA, and every time an organism reproduces itself, then it will shuffle around its genetic material a little bit, it will mutate a bit. And so we will get new phenotypic characteristics, so new physical characteristics in the offspring that are a result of this mutation happening in the DNA. So you might recognize these creatures. We've got on the right-hand side a North American grey squirrel. On the left-hand side, we've got one of our native British red squirrels. And if we look at the map of the UK, since the North American grey squirrel, which is not a native species, was introduced a couple of hundred years ago, there's been a remarkable disappearance of our native red squirrel. And if we just look at the last 40 or so years, you can see that it's really declined even within that period, back to these very dark green zones. So I want to ask why you think that is. So it's a bit of audience participation. I realise I'm competing here with Saturday night television. So Paul, can you just put poll 1 up? Okay, so what's the major reason for the expansion of the grey squirrel territory and numbers and the reduction in red squirrel territory numbers? Is it that grey squirrels are more aggressive? Grey squirrels have mutated since arriving in the UK to enable them to be more successful. Grey squirrels have a much wider diet, so they can survive better than red squirrels in times of famine. None of the above, or I missed the last few minutes, it's Saturday night and I was pouring my first or my second G&T. If you could answer that now. I promise this is actually a seed lip and tonic. Okay, so I think Paul will launch the results. Okay, excellent. So, so we've got, okay, most people think that they've mutated since arriving in the UK to enable them to be more successful. One person was having a G&T, cheers. And we've got a range of other answers. It's actually a slightly trick question, and the correct answer is none of the above, or possibly that you missed the last few minutes. If you can close that, Paul, then I'll just move on. Thanks. And the answer to the question is a virus. So there's a virus called squirrelpox. And squirrelpox has been co-evolving with the North American grey squirrel for a very long time, to the extent that the squirrel and that virus have really come to a good relationship with one another. That virus doesn't cause much disease in the squirrel population, although it will spread very easily within them. However, when the grey squirrel was introduced to the UK, if the virus gets into red squirrels, they haven't been evolving with this virus, and it is almost 100% fatal in the red squirrels. So it's a really serious veterinary pathogen, and a very sad story, the red squirrel. But it demonstrates that viruses evolve too. So every time a virus replicates inside a cell, there's a small chance, well actually there's a reasonable chance that it will mutate. Viruses are the organisms that we know of with the highest mutation rate. And if that mutation confers a genetic advantage, then it will obviously proliferate. So as we go through evolutionary time, and I'm talking about multiple generations here of the virus and the host, but the virus obviously mutates faster because it has very short generation times of something like a day, whereas the host has probably got longer generation times. Certainly we're thinking about humans. So they develop an arsenal of weapons and protective systems. So the virus might have a molecule that comes in and it damages the host cell to its advantage. And the host cell will develop some sort of protective system. And as we go on, then the host will develop some better weapons to try and eliminate that virus. And the virus will develop its own protective system against those. And the host will get, even more sophisticated weapon systems. And the virus will develop other ways of either hiding in this case, or of throwing more weapons back at the host. But over this long evolutionary time, you've got several different possibilities for the outcome. So the first possibility is that the host kills the virus species. So you have a transient infection in the species, it doesn't really get very far, and the virus dies out. The next possibility, which we hope doesn't happen from our perspective, certainly, the virus kills the host species. So it's so successful in spreading, but it's so pathogenic that actually that host species dies out. But what commonly happens is that over time, the two species will co-evolve with one another, and they will actually adapt to one another so that the virus will be allowed to spread, but it won't cause very much disease. So you get a sort of truce in this arms race. So this brings along the concept of the natural reservoir. And a natural reservoir is a species in which this virus replicates and spreads, but it doesn't cause disease. So in the story of the two squirrels, then the grey squirrel is the natural reservoir for squirrelpox virus. So if you are a grey squirrel and you're moving, you want a bit of new territory, then you could either wage war on the local red squirrels, which is going to take quite a lot of time and effort, really, or you could just visit their feeder. And you could have a couple of peanuts, leave a bit of saliva lying around, and infect the local red squirrel population with the squirrel pox that you're carrying. So how do these natural reservoirs and the global travel situation that we have affect us as humans? So we have a situation a few years ago that I'm going to tell you about. Smallpox was the first virus to actually be, smallpox was a disease caused by variola virus, and that was the first virus to be eradicated worldwide through intervention, human intervention, through this vaccination campaign. Smallpox was a really horrible disease. The virus spread incredibly rapidly and successfully, and it had a high fatality rate, so some strains could cause up to 40% mortality. So after we managed to eradicate smallpox by vaccination, there are only two stocks of smallpox that we know about that are held worldwide. One is in the Center for Disease Control in the US in Atlanta. The other one is in Vector Labs in Russia in Novosibirsk. There is also a virus that causes a slightly similar pathology, quite similar pathology, called monkeypox. So this virus is known to circulate in monkeys in West Africa. If it transmits into humans, then it will cause this horrible sort of pustular rash that's like smallpox. It's not as, the mortality rate is lower than smallpox, and it doesn't spread very easily from human to human. So those are two major differences. So in 2003, this little girl in Iowa came down with something that began to resemble a smallpox or a monkeypox rash, which obviously was very concerning for public health officials. And then over the next week or so, there was a cluster of infections spread across these states in what I think is probably the American Midwest, and they infected 75 people. Many young people were affected, including children. The households hadn't had any contact with one another. The households had not travelled outside the US recently, and the virus did not transmit outside individual households. So Paul, can you launch poll 2? Because now I'm going to ask you who or what you think was responsible, what was going on here. So why was this virus appearing in these disparate populations in the US? So was it an accidental release from laboratory? Was it an act of bioterrorism? Had the patients all had close contact with monkeys recently? Had the patients all had close contact with local American animals recently? Or it's a conspiracy theory, it didn't really happen, and you're off in your next G&T. So if you can vote now. Okay, and we can have the results. Okay, yeah, brilliant. Okay, so lots of you have put the answer, the patients that had close contact with local American animals recently, and that's absolutely correct. Can you close that, Paul? Thanks. Okay, so what had actually happened was that the virus had come, there was a link between each of these clusters, each of these families, and pet shops. So they'd all been to pet shops recently, and they'd all bought a pet. which is a sort of prairie dog or a groundhog-like animal, and they're a type of rodent that are native to the US. Whilst these prairie dogs were being housed in a sort of pet holding facility, they were housed alongside a consignment of small rodents that came from West Africa. These are called rope squirrels, and these are Ghanaian dormice. And there was also another one called a Ghanaian giant rat. And it turns out that the natural reservoir of monkeypox virus is not monkeys, but it's these several types of West African rodent in which the virus circulates, but it doesn't cause any disease. So without this rare event happening, we would still not know what the natural reservoir of monkeypox was and where it comes from. So it can actually be very difficult to try and figure these things out. But you get the concept that viruses have often a natural reservoir. They can be bats, that can be rodents. There's lots of different species that reservoir can be. And if we then house those creatures, or we bring them into contact with something that is genetically a little bit similar to them, but has never seen the virus before, then the virus can sometimes infect these creatures, and it will replicate to quite high levels in them, and it can start to cause some disease. And it's a bit easier then for that level of virus, that really high amount of virus, to transmit into humans. That's just an example of some of the other fairy vectors that we might have. So this can happen in terms of pandemic influenza. That's how new pandemics start. Influenza A has a natural reservoir in waterfowl. These obviously fly long distances, they migrate, they come into contact on their migratory journey sometime with pigs and horses. And those are creatures that are more susceptible to these viruses than humans are. And within pigs and horses, the virus can replicate to higher levels. It can mutate a bit, it can cause a bit of disease, and then it can transmit to humans, although possibly not this one because he's asleep. But the concept is that viruses often transmit to humans from a reservoir via a vector species. We see disease when the virus is not fully adapted to its host, but just because we don't see any sign of disease, it doesn't mean there's no virus there. Often a virus is transmitted to humans from a natural reservoir via an intermediate sort of furry vector. So coming on to SARS coronavirus 2, that's the virus that causes coronavirus disease 19, COVID-19. There is a similar virus, it's not an identical virus, but there is a similar virus that circulates in a type of bat from East Asia. And so this resembles the natural reservoir. However, it's not similar enough to be the virus that has directly infected humans. And we're still looking for what this intermediate species might actually be. The key here is that if there is a natural reservoir present, we're unlikely to be able to eliminate the virus from humans because it will live in the natural reservoir and it will just come back again. So we need to bear that in mind for the future. So that brings me on to how viruses cause disease. And in this, the number of the invading army matters. So the number of viral particles that actually try to enter the host, if it's a very small number, they're unlikely to breach those defenses. If it's a very large number, then they might well breach those defenses. So you think about our immune system as a sort of castle structure. We've got multiple layers of defense there that are what we call innate. They're present all the time and they are ready to act on these invaders. So the more virus a person or an animal gets, it's very strongly correlated with the amount of disease that we then see in that individual. So the second way viruses cause disease is that they can directly damage host cells. So the viruses can replicate inside the host cells, and in order to spread, they can burst that host cell open. And some viruses, the disease that we see is caused by their direct damage to cells like this. So a good example of this, herpes simplex virus, it causes cold sores, and that's where the virus actually destroys cells. However, we can also see disease caused by viruses when the immune system selects the wrong weapons to throw at them. So if we consider some sort of mythical bad guys here, we've got the vampire, we've got the werewolf. we need to select very carefully the sort of weapon that we're going to try and eliminate these guys with, in order that we're going to do it properly. So if we throw silver bullets or fire silver bullets at a vampire, we're not actually going to kill the vampire, but we might cause quite a lot of collateral damage at the same time. So we have to be very careful. And the immune system has got a huge arsenal of different weapons that it can choose from. And it has to try and identify what the bad guy is, what the invading pathogen is, and select the appropriate weapons and the appropriate amounts to throw at them. And you're all my witnesses that if I'm just blacklisted by MI5 for Googling every weapon known, then please, come to my defense. So another example of that, you might recognize these guys. These are the White Walkers from Game of Thrones. If we're trying to eliminate the White Walkers, then we can do that very directly if we use dragon glass. Or if we happen to have a handy dragon lying around, we might just throw dragon fire at all of them. And this is obviously a bit of a more drastic option, but it would work. If we have a rodent infestation, then it's a bit, it's a bit of a problem. We really want to try and get rid of it, but we could probably live with it if we had to. So what we probably wouldn't do is employ our handy dragon to try and deal with a rodent infestation. It would work to keep the rodents in check, but they can probably live underground. They can probably, keep there in small numbers, come back every now and then we just have to, burn the house down again with dragon fire. So we really want to try and, the immune system really wants to try and direct the appropriate weapon at the pathogen. But viruses can make that really complicated because they can actually fool the immune system into choosing the inappropriate weapon. And they can do that by hiding from them, by looking slightly different. And so I think about this as a sort of sheep in wolf's clothing. So the classic viral example of this is hepatitis B, where hepatitis B virus itself, it doesn't do a huge amount of damage to our cells. We could probably live with it. What does the damage is our immune system, which is really trying to throw a lot of weapons at hepatitis B virus to try and get rid of it. But what it actually does is just damage our own cells. And so we end up with a lot of liver damage, or we can do in hepatitis B infection. So the immune system's, its first job is to detect pathogens soon after they enter the body and to call for aid. So I'm just showing you here the beacons of Gondor. And so inside a cell, we've got these early warning systems. And when they detect that something looks a bit fishy and it shouldn't really be there, then they will signal to the inside of the cell, to other parts of the body, and they will select and recruit the specific armies and the weapons that they think they need. And those armies and weapons will hopefully come to the defense of the cell in trouble. However, if the virus has fooled the immune system into selecting some inappropriate weapons or throwing too much firepower, which is unnecessary, then we can get a lot of immune damage. And sometimes the immune system can't actually tell when it's even defeated the enemy because there's so much damage there. So this is what's happening in COVID-19, in this disease. So at the start of the infection, we get this viral replication, and the illness that we see is linked to a sort of initially appropriate immune response. We get a fever sometimes, and this is the body trying to eliminate that virus. But as we go along, sometimes the virus will be eliminated, and sometimes it will actually be replicating at quite high levels, and sometimes the immune system will just mistake it for something that's a bit more dangerous than it is. It will select the wrong weapons to throw at it. And then the really serious complications of this disease come from our own inflammatory response. So they're not actually caused by the virus itself, they're caused by our own immune system, our own armies overreacting. So if we go back to the situation of Lord of the Rings, Rohan and Gondor, then what we should do in the case of a sheep in wolf's clothing or a sheep in orc's clothing would actually be to say, okay, guys, I think you don't have 10,000 orcs running around here. You've got sheep. You might have a bit of overgrazing. This might be a bit of a problem for a couple of years, but actually, I think you can deal with it. And that's what these new drugs do that have been recently found to work and been approved for clinical use. So they really just calm down the immune system at the point when it's overreacting and it's trying to throw some really inappropriate weapons at this viral infection. So for SARS coronavirus 2, that means that we can actually use two different types of drugs, and we can develop two different types of drugs against it. So if we catch an infection really early on, then we might expect antiviral drugs to be really effective, or we would expect them, if they work, to be really effective at this stage. If we use antiviral drugs at a stage when there's no virus left, then they're probably not going to be effective. At that point, we need anti-inflammatory drugs. So there's a sort of a blessing and a curse here, that we have two different types of drugs that we can develop, and that might give us an extra arsenal with which to combat this disease, but we need to be careful that we're using them appropriately at the right time in somebody's infection. So that brings me on to how viruses spread. So they largely spread because of the cells that they infect. So some types of virus, like noroviruses, they cause winter vomiting disease. They infect the cells of the gastrointestinal tract, and that's also how they spread. Whereas coronaviruses, influenza, infect the cells of the respiratory tract, so they largely spread by things like coughing and sneezing. As you can see, coughs and sneezes can actually carry particles really quite a long way. And they can also, we think, spread in aerosols. Certainly some kinds of virus can, and there's circumstantial evidence that SARS coronavirus 2 might be able to spread in very, very small particles. So there's something that's been really talked about in the press a lot in the last couple of months, which is called the R number. This is to do with the measurement of how the virus is spreading, how easily it's spreading in the human population. And the basic reproduction value, what's called the R naught, is defined as how many people are infected per confirmed case, or per case of the virus, in a completely immune naive population, so a population that hasn't, none of whom have seen this virus before, none of whom have been exposed to it. So there are some viruses that are extremely easy to spread. So measles has the highest spreadability that we know of. Smallpox, variola virus had an R naught of about 7 people per infected person, so it's spreading extremely easily. The recent outbreaks of Ebola were much lower at 1.7, and MERS, which is another coronavirus, was below 1, so it died. Whenever it causes a new cluster of infections, it tends to die out fairly quickly. So the R value or the R naught for this virus is somewhere around this, estimated usually at around 2.5 to 4. So it's really quite easy to spread the virus if we don't take public health measures. There's an interesting story which is in a Lancet publication now, which was written by a respiratory physician who was on board this ship early on in the pandemic. And there were about 200 people on board this ship, and they took very appropriate measures to lock down as soon as they suspected that they might have a case of COVID-19 on board. They gave everybody masks, everyone was wearing PPE, people were confined to their cabins as much as possible. And they followed up all the passengers very, very closely. And interestingly, was more than half the passengers, about 60%, who became infected with the virus at some point from an initial 1 or 2 infections that were detected on board that ship. And 81% of those cases were asymptomatic. That's higher than estimates in the general population. And also the people on board this cruise ship tended to be of older age, and usually there's evidence to suggest that we get more asymptomatic infection in younger age groups, not in older age groups, really. So that was quite a surprising finding. And one hypothesis that's come out of this is that although it's very difficult to protect yourself from an infection, to prevent infection from occurring in a situation like this on a cruise ship, the fact that everybody was wearing masks probably meant that everyone was exposed to lower levels of the virus, and they were taking appropriate social distancing measures as well, which just takes me back to what I was saying about the number of the invading army there mattering. It's possible and probable that the greater number of viral particles you are exposed to, there's a correlation with the amount of disease that you might get. So that's my argument for wearing masks as much as possible. Obviously, there's a big problem with COVID-19 and its spread, and it's that it's largely caused actually by people who don't have symptoms. So the highest amount of virus appears to be shedding from people in about the 2 days before they start showing symptoms. Obviously people can also be completely asymptomatic and never show symptoms. Public health measures might not just prevent infection and spread of the virus, but they can also lessen the severity of the disease that's caused. So what works in terms of public health? We know from the HIV pandemic that we need to listen to science, act fast, and respect human nature. And I'm just going to speed up a little bit through this because I realise it's GMT time on a Saturday and I don't want to keep everybody from that. But we know from the South African response, which was to not listen to the scientists and say HIV was actually a disease of poverty, that we had a tragedy that happened in South Africa with the HIV pandemic and it becoming the highest rates in the world. There was another country here, Uganda, in which you can see it managed to control that infection very effectively. They did that by a very clear public health message. And that public health message included a recognition that some people were going to have a lapse of human nature anyway. And the public health campaign was very clear to include that. And very sadly, they've taken that out of the public health campaign in recent years, and HIV has spread again in Uganda. So today, sub-Saharan Africa is the epicenter of that epidemic. We know from the initial stages of the HIV campaign that, or the anti-HIV campaign, that it's less effective to demonize people because they're then less likely to go and get tested. If they're tested, they know they have the virus, then they can work out how not to spread it more easily. And so now we have treatment against HIV. It's much easier to spread the message for people to be tested and not to demonize people. We can also use drugs to prevent the spread of virus. This has been done incredibly well with HIV. It's called pre-exposure prophylaxis. And so if, for example, you are part of a couple, one of you has HIV and the other one of you doesn't, and you want to conceive a child naturally, then the HIV negative person can actually take this medication as well. And that's very effective in preventing HIV transmission. So something people often don't think about is that it's not just vaccines that will prevent the spread of disease, we can also use drugs to do that. Vaccination, however, is obviously an incredibly important tool worldwide. This is Edward Jenner. He's doing something completely unethical by inoculating his gardener's son with some deadly virus. However, he did also give him a trial vaccine a week or two before, which was cowpox virus. And luckily, this boy was protected, and that began the smallpox campaign, vaccination campaign. So vaccines work by training the immune system to recognize the invader and having the right tools and weapons ready to throw at it very quickly. One of the ways that that most vaccines are successful or unsuccessful is whether they can make the right sort of antibody against the virus. So these antibodies, they will, what we call neutralize, they will bind to the outside of the virus particle and stop it from talking to that host cell, so it can't get into the host cell anymore. Some types of antibody, however, will bind to the virus, but they won't actually neutralize it. So it can still talk to the host cell, it can still get into it, and they're not very useful. So for influenza, we know we can make great neutralizing antibodies. That's why influenza vaccination is so successful. For HIV, we can't make them. So it depends. If we can make these sorts of antibodies, it becomes much easier to vaccinate. The good news is that last week, one company released data to suggest that they've made a therapeutic neutralizing antibody that works in an outpatient setting on coronavirus patients. So that's incredibly good news. The other question to ask is whether the vaccine can elicit any detrimental responses. What we don't want it to do is to do something that makes the situation worse. So very occasionally, the virus can bind, you can have antibodies that bind to the virus, they don't neutralize it, and actually they make it easier for this virus particle to get into the cells it's trying to get into. And that happens with dengue. And it's really complicated vaccination development for dengue. There's also some publications that suggest that this can happen with other coronaviruses. So the vaccine development is going to have to be very careful to make sure that this is something that's not happening, because we obviously don't want to protect 95% of the population and make it far worse in the other 5%. So we need to achieve herd immunity. The question is what means that we can achieve it through vaccination. The more effective the vaccine, the fewer of the percentage of the population that actually needs to have it in order for herd immunity to be effective. And obviously we have some people in our society who rally against the use of vaccines, which makes life quite difficult, particularly with the use of social media at the moment. There's other people in our society who try to promote vaccination, but try to do it in a real hurry. And this can also be particularly unhelpful, because we really need to make sure that vaccines are very safe and very effective before they go into a human population. So in summary, we need to work out how this virus can transmit into humans. We have vaccine and drug development ongoing. In my opinion, they both look like they're possible, but they're unlikely to be completely straightforward. As these drugs and vaccines are becoming available or improving, we're going to need to prevent viral spread. And doing this effectively requires either strict impositions on our lifestyle, or it requires an effective screening program. In my opinion, using more asymptomatic screening than has been done, and effective contact tracing and isolation of contacts. We also know that social distancing not only lowers the spread of disease, but it's also likely to lessen the dose of virus somebody gets, which in my opinion is probably very important. So what do I work on? Well, I work on how these sorts of virus, which have RNA as their genome instead of DNA, have that genome structured. So this is our structural model of HIV, of some of the HIV genome. And what we do is we try to work out how those structures act as signals, how they control different things that are happening within the cell, so how they really help the virus. And then we try to work out how we can design drugs to design the important bits and important structures on this genome that might stop the virus from replicating. And we do that with HIV, dengue, and now with SARS coronavirus 2. So these are the people who have been involved in the work. And Andrew Lever works with me on all of this. He's been absolutely wonderful. He's also my partner listening downstairs. I do wonder if he was the 4% having the G&T. And Harriet Groom is our co-investigator to look at the SARS coronavirus 2 work. These are the Homertonians who have been through the lab or currently in the lab and have been a wonderful help in the various projects that we've been doing. Caden is currently working on SARS coronavirus 2. He's about to do that for his part 3 project. James worked with us last year on dengue. Ben has been working with us on HIV, and Aaron has been working with us on actually everything. And yeah, with that, I will hopefully take any questions.
Julia, thank you so much. That was an absolutely glorious romp through not only virology, but also Harry Potter, Game of Thrones. And yes, you've hit all the cultural buttons here, I think, for a Homertonian audience. So thank you very much. We have some questions coming through on the chat. I'm going to abuse my position as moderator, though, to ask one of my own first, which is, what do you think could possibly account for the huge variation in symptoms that people experience from this disease, from no symptoms whatsoever through to the most life-damaging?
That's a really good question, a very difficult one to answer. I think the most important thing to begin with is the dose of virus that somebody gets. The reason I say that is not only from what we know about animal studies from other viruses, but that healthcare workers have really had some very nasty symptoms and disease. And I think the people who are likely to get exposed to the higher amounts of virus are often the ones that are coming down with a greater amount of disease. There are thoughts about how the virus enters the cell, the receptors, and the different proteins that it uses, and the sort of demographic of the population that this might that might be affected by this. So there's a thought that perhaps men are expressing more of the receptor that this virus uses to get into cells. In terms of the impact on those with obesity, type 2 diabetes, who are more susceptible to disease, to more serious disease from this virus, they also tend to have a greater amount of inflammation in their body. So obesity is linked with a sort of slightly overactive immune system in that respect. So I think they're probably a little bit predisposed in that sense. Yeah, and other than that, I just think there are so many different factors that are involved. Yeah.
Okay, thank you. Right, moving on to actual questions that actual people have asked. Caroline has asked, I have read, says Caroline, that this is a once in 100 years event, but listening to all the ways that viruses can mutate, that might seem optimistic. What's your view as to the frequency with which we'll see this?
Yeah, I think that we will see this sort of pandemic more frequently than we have done in the past, because we are coming into contact with animals more than we have done in the past. We are encroaching on their sort of their natural territory, and we're living in higher densities. So there's a real environmental argument here, and I heard an environmental economist make this argument on Channel 4 News last week. I was really very much in agreement with him, that the more we can separate human activity with the environment that other species actually live in, the better.
Okay, thank you. Question from Harry, who asks, do the drugs that can act to calm down the immune system, do they pose risks for reducing the immune system's ability to fight other diseases?
Yeah, absolutely, they can. So hopefully, if you're at the point where you're having to take these diseases, you are in a pretty, these drugs, you're in a pretty sheltered environment, and you're less likely to then be exposed to other things. Our immune system it's geared to be able to respond very well to one type of pathogen at any one time. So if we take down its ability, if we dampen its ability to do that, then yes, we will be more susceptible to other diseases. But also, if we're infected with a different pathogen at the same time, then that can also make us more susceptible to disease from COVID-19, because the immune system's less good at controlling its replication to begin with.
Great, thank you. Jackie asks, are those people who have immune systems which consistently overreact to perceived threats, for example, MS, are they more at risk, or does it depend on whether they're taking immunosuppressant drugs?
That's a very good question, and I don't know the answer to that. I do have a medical opinion downstairs, I think. He may make his way up and answer that question if I'm lucky. Yeah, perhaps.
That's right.
Yeah, do you want to move on to the next question?
While we're sourcing an answer to that. Will Palmer is saying, asking.
Hello, Will.
Yes, he begins.
No, you were the GP person.
He begins his question with, "Hi, Julia. I think you may know each other. Do you know if, from the study of COVID, we've learnt anything new about other known viruses and pathologies? Can COVID be an insight, a way into understanding?"
Well, from my perspective, there's an interesting thought that the RNA, because COVID has this incredibly long genome, it's 30 kilobases, and that's the longest RNA genome that we know of in a virus. And often the immune system would recognize that straight away. And COVID-19 also has some very strong RNA structures within it, and I know Will understand this because he's ex-Bionatsky. But the COVID-19 is very good at stopping the immune system from recognizing that. And these very strong RNA structures within it are allowing the viral RNA to persist in an unusual manner. So I guess the answer is not yet. I don't think it's taught us anything concrete yet, but I really think it's going to.
Great, thank you. Any progress from your medical advice?
Yeah, here we go. Medical advice.
Hi, Andrew.
Hi. Yeah.
Hello. Thank you for joining the panel. Welcome. So I think that the question that Julia thought you might have insight into is whether conditions which consistently overreact to perceived threats, and multiple sclerosis was mentioned as an example, would those conditions make a patient more at risk, or is it a question of whether they're taking immunosuppressant drugs? Does that have an effect on the answer?
Really great question, and the answer is that no one really knows the full answer to that. If you're taking immunosuppressant drugs, obviously you're more prone to get the infection and not overreact. But in people who've got an overreaction, such as in multiple sclerosis, which was mentioned, it tends to be focused on one particular thing. So people with multiple sclerosis don't necessarily get rheumatoid arthritis, for example. Their overreaction tends to be focused in one area. And so that means they probably wouldn't necessarily get an overreaction to an external thing like a viral pathogen.
Great, thank you. While we've got you both on camera, before you escape to that G&T.
Third G&T.
It was you, yeah. Can you tell us something about what happened to you and your lab when lockdown hit? Presumably you've spoken a little bit about this, but I wondered if we could explore that in another couple of minutes. You will have had some early warning of the fact that this Wuhan wet market was incubating something that could be of global significance. So you presumably were busy in your laboratory working on HIV and FIV and dengue, but with half an eyebrow cocked at Wuhan. Can you tell us how you took the decision, or was the decision taken for you, to pivot your research and to start working on COVID?
So we were very keen to work on COVID because it was becoming more and more evident that this was going to be a virus that was going to affect a lot of people, and that was going to spread worldwide. And we work on several different viruses already in the lab, and our expertise is on RNA, and coronaviruses are also RNA viruses. So we have a lot of tools in the lab to deploy to study this sort of virus, and there were actually emails going out from the RNA community saying, which is a worldwide community of scientists, talking about this and talking about the need to switch research to this coronavirus, and to all rally together and collaborate more than we usually do. I mean, we usually do collaborate quite a lot anyway, but it was a very cohesive community of scientists with a lot of goodwill to try and do what we could to understand it. Obviously, that takes quite a lot of time to do that. So we did start. Well, we started in March, I think. Yeah. But then we were told by the university on shutdown that we were not allowed to work on anything other than SARS-CoV-2. So that meant that actually the whole lab switched their focus to it. And we then worked as two teams within the lab to look at two different aspects of the virus. And it was really rewarding for us to do, because normally we work on our own little projects. And we might work on them in groups of 2 or 3, and so to have one focus for about 12 people was really interesting.
It was very cohesive.
Yeah. now we obviously we're back to now trying to do both things at once, and so that's why we've recruited Caden, our Homerton student, who will be helping to do some of the lab work associated with the SARS-CoV-2.
Fantastic, thank you. I think we're about ready to wrap up. I'll ask if there are any final questions. Please type them quickly in the chat if so. But in anticipation of that happening or not happening, I'll ask Verity's question, which is there any particular type of gin that you'd recommend during this time?
I really like West Wind's The Cutlass. That's my favourite gin.
Great. Okay, thank you. Yes, we have had a couple more questions come in. You might not wish to answer this, but we all have our opinions. But to what extent has politics influenced collaboration?
Influenced collaboration?
Not quite sure what is. It struck me that I'm not the questioner, but it struck me how much of what we're hearing from the news about possible breakthroughs, whether that's new vaccines or treatments, has a very sort of nationalistic flavour to it. That the UK is talking about the Oxford vaccine almost exclusively as the thing that might get us out of this, and presumably there are other vaccine candidates in other countries that don't get any coverage here because they're not ours. Am I right to think that the scientific community is rather more joined up internationally in collaborating to address this, or are these national walls affecting science?
No, I think that the vaccine development is fairly. There's a commercial aspect to that as well. So I think individual groups and their commercial partners are probably relatively territorial about that. I think we talk about the Oxford vaccine a lot in the UK because we've obviously signed up to trying to get 100 million doses of it. So if that vaccine works, then that's probably our first port of call. I would also say that I think that Oxford vaccine is well designed, that it is it's using a sort of vector that is very good at provoking a strong antiviral a general strong antiviral immune response. It brings in a type of cells called T cells, which we also need against a viral infection. And so I think that's a very sensible way to design a vaccine against this disease. So
thank you. So yeah, we've got a couple of questions coming in. Let's see if we can answer those before our hour is up. So once Holly asks, once you've had COVID-19, do we yet know how likely you are or whether it's possible to get it again and pass it on to others?
We at least know that it is possible to get it again because it has happened. There have been recorded cases of that happening. It doesn't seem to be happening with high prevalence yet. So in my mind, it would be likely that you would have some degree of immunity for a number of months at least. But with other coronaviruses, you are then susceptible to another infection, often after a relatively short period of time. So it's something that we definitely shouldn't rely on people being protected if they've already had an infection, because they might not be. Is that fair?
Yeah, thank you. Very specific question on taking vitamin D, which has been touted as being helpful. Is it true that vitamin D can stimulate T cells and therefore help? Yeah,
vitamin D tends to stimulate something called regulatory T cells. So we need vitamin D in order to have a healthy balance of all of our different types of T cells, and those regulatory T cells in particular are they're important at controlling when you're having the wrong sort of immune response to a pathogen. So although I don't think there's a definitive answer about that, if I were you, I'd take some vitamin D because probably wouldn't do any harm, and it can certainly protect you against lots of things and make you healthier generally. You think lots of the UK population are vitamin D deficient over the winter anyway, don't you?
They are, yeah. Vitamin D should be almost routine for everyone over the winter.
Brilliant. Everybody in the north of England, at least, and points north. Question from Harry Gooch, who asks, would love to hear your stance on why so many, brackets, non-scientists, are keen to dismiss the virus. It's just the flu, it's not as deadly as the flu, and so on and so forth. Why is it that we are, as a population, we tend to respond by denial? She says this might be a sociology, psychology question rather than a biological one, but the comparison to the South African reaction to AIDS got me thinking.
Yeah, Harry, that's a really good question. I just don't know the answer to it. It's really frustrating. Obviously, we're all finding ourselves in a situation that we haven't been in before. As scientists, we're not really used to communicating with the wider public on a frequent level, and I think that doesn't help because we're having to respond so quickly at the moment. But yeah, I think there's some very worrying opinions, some things circulating on social media that are really not helping, and some political decisions that are really not helping as well. Yeah, it's a great question. I can't answer it. Can you answer it?
No, the South African situation was slightly different in that the South African authorities were very upset that people were thinking that the sexual behaviour in South Africa was the same as the gay population in the west and east coasts of USA. And so they felt this was an insult to them, and so they reacted in trying to make a case that HIV wasn't caused by sexual transmission. But I think Julia's otherwise absolutely right. One of the problems is we're giving people an unpopular message: stay at home, don't go out and have fun.
There are no more questions coming through. I will end then by asking one final question while we're on the subject of public health advice. What is the most effective thing that the people listening to this webinar should do to protect themselves and to protect other people?
You want one thing?
No, you can list things with an error bar if you like.
Yeah, so the first thing that I would do is consider your daily interactions. If you can switch things at the moment to an online interaction rather than a face-to-face interaction, and you're not going to lose a huge amount by doing it, then I would do that because the virus is spreading pretty quickly at the moment. And so everything that we can do to just prevent that we should. So, the message to work from home, if you can, is absolutely the right message. I think we should have that for a lot longer. Secondly, be a good citizen and, wear a mask and try to persuade others around you to do that sort of thing. Maintain social distancing and try to be a voice of reason within your community about the need for good public health measures at the moment, in order that we can try and really contain this to the extent that we can get a better screening program up and running, including asymptomatic screening, because then we might actually be able to release the curbs on daily life, or a lot of them, and still maintain a very low infection rate. So that would be my hope for the next 6 months.
Great, thank you so much. Thank you, Julia, for a fabulous presentation. Really, really engaging. Thank you so much. Thank you, Andrew, for joining the panel for the Q&A. Massively appreciated. And thank you to our alumni participants for some really great questions and for engaging so well.
Oh, and the other thing I wanted to say, Matthew, sorry, was if we have any teachers out there, anybody connected with schools, I'm very happy to deliver a version of this seminar without the gym references, which can be either a scientific or a non-scientific version. So just let me know if you would like me to do that. I can do some outreach.
That's a really fantastic offer. Thank you. So yes, if any of you would like Julia to talk online to schools that you're involved with or other groups, then I think the best thing to do is if you email alumni@homerton.cam.ac.uk, then we can pass that message on. So that's a really kind offer. Thank you very much, Julie. Thank you, everybody. So stay safe, wash your hands, and cheer for Homerton, because we've got some really good people, both our academics and, as Julia has pointed out, students who are working directly on this as researchers, as well as, of course, the college doing everything that it can possibly do to keep our own students safe and engaged and informed as the academic year starts. So thank you all for your participation. Thank you for your support. Have a very good evening.
Yes.
And I hope you've all remembered that gin recommendation. Thank you again. Take care. Bye-bye.
Bye.